All right, so welcome to the ATUM Science Seminar. This time we've got a really exciting guest from Eli Lilly. Lucas will tell us about how we're dealing with immunogenicity. And this has really become one of the key drivers for the protein pharmaceutical space and how we're going to take on these big challenges. And on the next slide. So just to showcase the differences here, we have looked at the top 10 drugs in the US over the last from 10 to 15 to 2019. And just to see this avalanche, is really the sea change of how the pharmaceutical business is focusing and on really replacing chemistry with biology. So here you see in 2010, 10 of the top 10 drugs were all small molecules. And just go 5 years later, this entire multi-billion-dollar industry has changed from where the top seven of the top 10 are now protein biologics. And just adding another 4 years, we don't have the 2020 data yet, but again, just clearly this is reinforcing the emphasis on proteins and biologics as overtaking the pharmaceutical industry. And of course, that has all sorts of implications on production, manufacturing, safety profiles, and all these different things. And of course, a majority of these things, these biologics today are regular antibodies, but increasingly we see bispecific, trispecifics, FABs, as well as gene therapy, cell-based therapies, and so on, really coming at the front across the entire pharmaceutical industry. Now, biologics are fantastic molecules, and because at the end of the day, we are all biologics, and it really changes the tools and the specificities of as we can engineer these drugs. So biologics, of course, are fantastic because they're target specific. The manufacturing process can be really standardized because at the end of the day, it's DNA and it's RNA and it's protein. It's very defined, limited side effects. And if you just look at the statistics, there are much higher success rates in clinical trials than do small molecules. But one of the core issues I think the industry is dealing with, and really what I'm struggling with at this point, is how do we deal with immunogenic response? How do we ensure that we minimize the immune system from attacking these molecules? And it's been really interesting to see how this field has rapidly evolved, and we're starting to get to a point now where we computationally and experimentally can really assess what are immunogenic molecules and which ones are not. And today we are fortunate to have Luke here come and talk from Eli Lilly and publish a beautiful paper here where they did, and I think you'll really appreciate how he was able to turn that problem kind of upside down and come from a different angle and looking at how do we see the immunogenic responses and how do we deal with that. So Lucas, I hand it over to you to take it from here and tell us this beautiful story you have.
Thank you.
Stop sharing.
Klaus. Oops.
And Lucas is currently at Eli Lilly, and it's been prior to that was at Boehringer Ingelheim for many, many years. He's got his PhD back at the University of Illinois in Urbana-Champaign many years ago, and has really been, ever since the very beginning, been focusing around immunogenic response and how the cells respond to these threats to the system. So no, it's going to be a beautiful story.
Thank you, Klaus. I appreciate the introduction. So the topic for today is I'm going to be talking about deimmunization of high-risk biotherapeutics. And what's different is we're going to actually use cynomolgus macaque as a surrogate for human. So as Klaus mentioned, Immunogenicity is a significant concern for biologics development. And, unfortunately, consequences of ADA vary a lot between, both molecules and individuals. Of course, presence of ADA is never desired. Some of the potential problems are in safety. So you can get hypersensitivity or anaphylactic reactions. So, one notable example that comes to mind is cetuximab and the glycosylation, and how a certain cohort of patients in the southeast part of the United States actually had an anaphylactic reaction because they were bit by a tick that had a very similar looking glycosylation pattern. Of course, you can neutralize the activity of endogenous counterpart. And this is, most thought of in the EPREX case in EPO, where an uncoated rubber stopper leachates actually provided danger signals to EPO and caused pure red cell aplasia in the patients that were taking it. You can, of course, get immune complex formation, which can lead to deposition of different organs and can lead to, of course, problems. Of course, efficacy could be affected. So you can neutralize the activity of the therapeutic protein, so you get neutralizing antibodies. There are other cases where ADA could bind, so such smaller things like peptide therapeutics, and they can actually extend or reduce the half-life, of course. And of course, you can also have the biodistribution being affected. Most commonly, though, immunogenicity basically reduces the exposure or the half-life of the antibody. And this can, of course, influence dosing. Then you may have to try to dose dose past the ADA. So there are a lot of factors that determine immunogenicity risk, many of them actually, and so they can most commonly be broken down into three main categories. Those here on the left that are associated with the product. So these are directly related from the product. So this could be sequence, manufacturing, and so one of the things that affects it is this thing called humanness. So how human does your biotherapeutic actually look in the sequence homology, essentially. We no longer call that at Lilly. We actually call it a rarity index. So we have some in silico tools where we actually look and see how rare are certain substitutions at different areas of a sequence. There's, of course, very classical immunogenicity, but T-cell epitopes identification. So there's different tools that can be used to identify these. Of course, the presence of these that are immunogenic, not the tolerogenic ones, it could be problematic for your biotherapeutic. There's, of course, B-cell epitopes. What this means and refers to is this is the cell surface epitope on your protein that the B-cell receptor actually recognizes, which will then be the same epitope for the high affinity antibodies that are secreted by those B-cells. Of course, if your molecule doesn't have very good biophysical properties, it can form aggregates. Fragments can form as well, and the aggregates are known to basically stimulate innate immune receptors, which can drive further immune responses. There's, of course, different types of post-translational modifications. So, glycosylations is an example. Things that can happen to the molecule, such as an oxidation, can increase aggregation as well. Pharmacokinetics, immune modulation. So if you have, these are kind of associated with the product and the biology of disease, but basically, oftentimes a faster PK could be indicative of some off-target binding. This, of course, when you saturate the target, of course. And so this can lead to higher uptake by antigen-presenting cells. Of course, if your biotherapeutic is an immune modulator, this can help in some cases. As I mentioned with the EPREX case here, you have some impurities that can be present in the formulation. Potentially some excipients can cause a little bit of problems. I know, as an example, sometimes people prefer not to use citrate, although it's not necessarily associated with immunogenicity. It can be caused with injection site pain. And I mentioned non-specific binding could also lead to increased uptake by antigen-presenting cells. There are those factors that are associated with the biology or the disease, such as the route of administration. Oftentimes, not always the case. It's generally thought of that by going by IV, and for biology disease here being more so for oncology, you'll tend to be less immunogenic since your molecule distributes broadly in circulation initially and isn't subject to staying at a higher concentration at an injection site for a little while, which can then be taken up by antigen-presenting cells in the skin. However, I've seen cases where the opposite is true. Actually, subcutaneous route was less immunogenic than IV. So this will be dependent on the molecule. Of course, dose levels. So very small doses, very low doses could probably lead to insufficient antigen presentation by antigen presenting cells. So you just don't have sufficient amount of presentation of the foreign antigen and stimulation of the relevant T cells. Of course, on the higher end of things, a very large bolus, such as when you're trying to dose past can sometimes, in some cases, lead to some immune tolerance. There seems to be a sweet spot for generating the highest levels of ADA, and it unfortunately seems to be right where we dose the dose levels that we use for normal biologics. Dosing frequency get affected. It's often thought that less frequent dosing can lead to higher immune responses. So if you dose once and you don't have to dose until 6 months later, that potentially could be more immunogenic. So although it's not always the case. Indication. So if you have an acute disease and you need one dose basically. So let's say in the case of anti-COVID antibodies, as an example, you may only need to get that treatment once, while a certain fraction of patients will develop an ADA response. There won't be another dose, so there isn't a boost or like a vaccine type of a response to drive a higher affinity ADA. Chronic indications, of course, the more times you have to dose something, you increase the risk of developing ADA. Target, this is a hotly debated one. It's often thought of that soluble targets potentially, so when an antibody to a soluble target potentially to lower rates of ADA, and then targeting cell surface proteins is higher risk. And it's often thought of that the cell surface targets and antigen presenting cells represent the highest risk. Although specifically for this particular study, you'll see we are targeting a cell surface target on antigen presenting cells. So it's on membrane TNF on dendritic cells. And you'll see that that's not always the case because we have molecules that aren't immunogenic at all. There are those that are associated with the patient, so their MHC background. So for anyone that's used an in silico prediction algorithm tool, such as EpiVax or the IEDB tools, as an example, you could see that there are certain MHC backgrounds where the peptides are predicted to bind more strongly to the MHC. So there can be patients that have a lower risk based on their MHC haplotype. Of course, tolerance to protein. So if you've been exposed to something a lot, so I like to use the example. So I think I want to say it's EPO is also one of those proteins that has a terrible in silico score. Yet most people don't have immunogenicity to it. So there's a lot of proteins in your body actually that have bad in silico scores, but your body makes them all the time. It's been making them since birth, and so there was a process of tolerance that happened as your immune system was developing. And so if you've seen the protein a lot, generally speaking, you'll develop tolerance to it more than not. Age can be another factor. So very young patients with less mature immune systems or very old patients can have slightly dampened immune responses. Immune competence, of course, if you're immunocompromised and you're taking chemotherapy, things like that, then you'll generally have a lower immunogenicity risk. And of course, I like to put this in unknown factors. We don't know everything about immunogenicity. I'm sure in the coming years, there's going to be a lot more that we're going to learn. And so I highlight that as unknown factors. So I put blue around some of the things that we can actually use in silico tools to help us predict immunogenicity risk. But again, these are just risk factors. So again, I mentioned you can have something that has a very bad in silico score, but if it doesn't bind anything, has very good biophysical properties, very good PK, it's just going to be taken up very slowly and eliminated. And so you're not going to have this big bolus of antigen present at one time. And this is one of the reasons why we actually see that not everything is foreign proteins, not necessarily immunogenic, even in mice or even in monkeys. So some of the important questions that this study tried to address is, so if you remove the hotspots predicted by these in silico tools, do you actually reduce immunogenicity? And so it seems kind of, mundane or obvious, that we're asking this question, but one of the reasons why is when you just trace back and you go back into the literature and you look at some of these publications where they've actually reduced the in silico hotspots, most oftentimes multiple factors were reduced at the same time. And so oftentimes, so as an example, they can reduce target binding, alter biophysical properties while they're removing the hotspots. So one common example, I'm going to show you on the next slide, but it's essentially a single point mutation in Humira, where you can actually, it's not immunogenic at all, but two things were changed at the same time, both removal of an in silico hotspot and completely eliminating target binding. So we feel that there was insufficient evidence or very little evidence in vivo where only the sequence quality was altered, maintaining some of the other properties the same. So the other important thing is there a way to test, validate any of these tools or predictions, in an appropriate animal model without going into human patients? Transgenic mice are, even though they may have some human genetics in them, it's not all human, as an example. And some of the tools that are used, T cell proliferation assays, certain molecules that modulate immune responses actually test clean in some of those assays. Not to mention they're not necessarily a perfect system. They don't really replicate what happens in your lymphoid tissue. It's basically just activated cells that you're mixing with your biologic, and so you generate some false positive and some false negatives in those particular assays. And so we wanted to do something in vivo into an appropriate animal model. And so our study basically tries to reduce these in silico hotspots while maintaining all the other risk factors the same as the parental molecule. Of course, it's not going to be exactly the same. It's a caveat here that you're not going to be able to have it be exactly identical. So we bet on using cynomolgus macaque as a surrogate for humans, and I kind of want to explain why we think it will work. So if you actually go to GenBank and you get the cynovariable regions of antibodies, they're up to 95% homologous to humans. The range is typically 80 to 95% as compared to the closest human germ lines. This is actually pretty high. Many of them were in the low 90s. So many biologics, if you inject them into monkeys, actually don't elicit any immunogenicity, even if they bind target. So it's we also felt that it was a low degree of complexity to cynoize a variable region. So, just a handful of mutations or positions would need to be mutated to make the molecule be more cyno. Next one is a little bit more complicated. So cyno-MHC alleles, shockingly, it's when you look at cynoproteins and you just compare a target that you're going after, like let's TNF as an example, you'll get very high homology between cyno and human. When you look at the MHCs, it's less homologous. So they're called MAFA, where they're called HLA in humans. They're also about up to 95% homology. I didn't see higher than 95% homology. Of course, sequences are still coming in. So the alpha chain of the class 2 MHC is about 80 to 95% is the range in the homology. Beta chain also can be up to 95%, but there were a few alleles where they were much lower homology in cyno. And this is going to be key here on strain usage that I'm going to talk about in the next slide. So it is strain specific. High homologies should be relatively predictive. The genetics are more complex in human and non-human primates than they are in something like a mouse or a rat. So we felt that it was more similar to humans and how the antigen would be processed basically and how the immune response would occur. And then there are publications and patents on how strong T cell epitopes are known to be promiscuous across multiple species, from mouse all the way up to human. You can think of Padre as a synthetic strong T cell epitope. There's also strong T cell epitopes present in other proteins that you can basically use these tools. So IDB as a mouse tool, they have a human tool as well. And you can look and you can see that if it's a strong T cell epitope, meaning it'll drive a high affinity ADA response, it will be promiscuous across multiple species. And so there's no precedent in the literature for using cyno for this particular purpose. So some of the other in silico tools that are used are the ones for predicting B cell epitopes, aggregation prone regions as an example, that those aren't species restricted. So that's just a fundamental property of that particular biologic that you have. And so we feel that the results obtained in cyno should have more translatability to humans. So essentially what we're using it is we're testing different strategies and using cyno as a surrogate for human. Try to validate these. So I think an important slide here is that not all strains of cynomolgus macaque are equivalent or suitable for immunogenicity studies. So there are two different types of cyno populations. There are those that are called isolated cyno populations, and these are known to not have any rhesus introgression or inbreeding basically. So these are more inbred, have smaller founder populations. These are typically found on some isolated islands like Mauritius, basically cyno. There was publication, Immunogenetics came out that said that they have identified only about 6 MHC haplotypes in Mauritius cyno, so quite inbred basically. Philippine strain had a little bit more, so they were around 20 haplotypes. While there are some in the non-isolated cyno that have less haplotypes, the Philippine ones also did not have any rhesus introgression. Now, when you look at certain proteins, rhesus appears to have just a little bit more homology to humans than what cyno does. So these types of cyno animals are basically good for controlled animal models. So they probably give you less variability since their genetics are less variable. And so these are good for something like you want to look at a PD study as an example. So these are likely to either over or under predict ADA translation in humans. There's examples where certain molecules, I'm not showing anything here, but we've used Mauritius and you didn't have ADA, and then you have your outbred strains and then you have ATA. So I personally would feel that these should not be used for any kind of immunogenicity or ADA translations for humans. There are other cyno populations, and these have rhesus introgression. So these are outbred, they have consistent crossbreeding hybridization. And this is just from some publications that are cited here in the bottom right, but that makes the haplotypes. So for Java is about 19, Vietnamese it's 13, so it's less than the Philippine, but they have rhesus introgression. Chinese were estimated to be at greater than 100. Sequences are still constantly coming in. Cambodian was listed to have greater than 100. So what we ended up doing is we were curious. We asked our suppliers, well, what strains do we have in our colony? And it turned out they said, oh, it's a mixture. So we said, well, what kind of mixture? And so what we ended up doing is we ended up basically sequencing our colony to see, what strains do we have. And it was estimated that we have greater than 236 haplotypes based on the sequencing of colony. We published this recently. These are similar to another publication that shows that those also have very large amount of haplotypes. So most breeding colonies use a combination of all these. They just call these Southeast Asian is what they call them. So if you ask, and it's just a mixture of them. And so we feel that this likely, because of the more complex, immune genetics, you could say, that you have better translation of the ADA results to diverse human populations. Unfortunately, these aren't as good for controlled animal model experiments, but you'll have similar variability as you will for humans. So if you do use these for PD, those results are more likely to reflect what you probably see in a human population. So I have this slide here to show that foreign sequences are not inherently immunogenic. And what this is here is there's a target binder, non-target binder, and these are basically an anti-TNF. And so there's a single point mutation. And this is what the PK looks like. And total IgG is what the PK assay was. And you could see that when you bind target, you slowly start losing exposure at about the 10-day mark. And it's completely lost in all of the animals by just after 504 hours. Whereas if you don't bind target, so there's a single point mutation, is the only thing that's different, you have exposure out 28 days of the in-life study. When we look at the titers on these, so the bottom here graph shows you have a decent high titer ADA in all of these animals. And we looked at the same thing in the ones that didn't generate any, no loss in exposure. And we went all the way out to 1,896 hours when we did not detect anything. So this molecule is just not immunogenic whatsoever. So what I'm going to show next is this unrelated molecule. So it's not related to these two molecules. So this is the only thing that it shares the same human IgG1 backbone, basically, as the binder molecule that was immunogenic. And when you look at this completely unrelated molecule, different germline families in the variable regions of this molecule, monkeys never been exposed to this molecule. And if you take the ADA from this particular study, so the bleeds from the immunogenic molecule, and you screen it against this unrelated molecule, you see that you actually have ADA to it. And so likely what's happening in a lot of these studies, and we've seen this using other examples too, other different types of formats, although there's not enough time to show all that data. But basically, you break tolerance to human IgG1. The human IgG1 is normally not immunogenic in monkeys. However, if the variable regions are immunogenic, they will break tolerance, and your monkeys will now react to human IgG1 to the FC. So we thought then, since we didn't want to take any chances here in the studies, that we'd use cyno-IgG1 and kappa light chain to help with the tolerance in the monkeys and focus our work just on the variable and changes in the variable regions. So we decided again to use cyno. So we took human antibodies. So starting compounds, we wanted to have something to be something that's been proven to be immunogenic, that we have data, previous data for, or data is available for non-human primates and in humans. So we chose adalimumab and golimumab. So these are mAbs with known ADA issues, although golimumab has a much lower rate of ADA in humans, but they bind the same target. High-risk target class is membrane TNF and antigen-presenting cells. AbbVie published a paper showing that this has an increased risk or increased amount of internalization on these cells. So then we used various engineering strategies that are based on in silico tools and the state of the art in humanization, deimmunization essentially, to deimmunize the molecules. And we try to preserve all the other properties. So the properties measure affinity, aggregation, melting temperature, just to focus on the sequence quality. And then all the compounds are tested in cyno using a single 5 mg per kg IV dose. So here are, and this is on human IgG1. So these are adalimumab and golimumab molecules that could be purchased basically. And you can see the exposure for adalimumab in cynomolgus macaque. You lose exposure after, there's nothing after 336 hours. It's just completely gone for these 3 animals. For golimumab, you see that 2 of the animals, you lose exposure after 336 hours, and then the other animals, you lose exposure later. And so looking at the titers, this is an ACE bridge assay. So because these are on cyno-IgG1, you have to use a little bit more complicated than a direct code assay that I showed in the previous example. And you see that there's ADA titers for both animals. And so the label ADA rates are for adalimumab between 1 and 26%, golimumab between 2 and 7%. And this is just taken from the web. And the higher range is usually listed for monotherapy. So this is basically where the patients aren't also taking methotrexate. Keep in mind that monkeys are not on methotrexate. And so, so they're basically getting monotherapy. And so, you know, while this is one example here, we've done other examples. The severity of the ADA is correlative with observations of human patients. This, along with the genetics being also highly homologous humans, suggests that cyno may be a suitable animal model. It's not perfect, of course. So next what we did is we took the variable regions, we put them on the basically make human cynochimerics, put them on the cyno-constant regions. And this again to provide tolerance spreading and hopefully reduce levels of ADA towards the variable regions, but then also to allow us to focus on just fixing the hotspots. So the sequences for cyno-IgG1 and IgK were available on imgt.org. They have actually the full antibody sequences for a lot of different species. So definitely germlining to increase the cyno content, hopefully improve the tolerance even more, and then deimmunization by the hotspot removal. And so these are just the chimerics. These aren't, deimmunized or germline molecules, but chimeric adalimumab and golimumab are actually less immunogenic in cyno. If you remember prior, we had no exposure after 336 hours for adalimumab on human IgG1, and you see just the presence of the cyno IgG1 actually gives you just a little bit of tolerance. And then for similar result for chimeric golimumab, you start seeing all of the animals now start getting exposure out to 504 hours, which is about 3 weeks. You see the titers are much lower. It appears to be clean, but it's not necessarily clean because you lose exposure. So there is some presence of clearing ADAs that are present at low titers still. So there definitely could be improvements since you don't have full exposure. Now remember, these are still binding. We did not change the variable regions. They're still binding membrane TNF. So we calculated affinity, of course, for cyno and human membrane TNF. So then we proceeded with germlining. And so these are some of the differences here. And you could see the top 2 is adalimumab, variable heavy region here with the closest human germline based on just doing like simple blasts. And then you can see what we found in GenBank is the closest cyno germline. And this is the designation here. And you can see very few differences here. And so that we were giving the option here of mutating. And then in the light chain, there was just a single site that we decided to give the option for. And these had similar homologies in cyno, these 2 particular cyno germlines. So we tried both of them essentially. Golimumab, you could see there's a little bit more differences. We did the same thing here. And how the screening funnel worked is we made shuffled essentially all the combinations of the variable kappa, variable heavy into a library, crossed them with each other, did small scale transfections to see what the titer was. And we were comparing this the whole time back to the parental. So the parental molecule was transfected as well as a control in every experiment. And then we get similar percent aggregate as the parental, similar affinity for TNF alpha, both human and cyno, similar Fab TMs. And then once you have several candidates, we would look for the molecules that the highest percent cyno or that appear to be just cleaner by other in silico measures. And then these were then put in for 2-liter transient expression and then put into some PK assays. We did this at the same time as the deimmunizations for the germline variants. We did the study at the same time, so we didn't wait for the results. There weren't that many differences for the germlining, so we thought it would be safe to go ahead and do deimmunization as well. So the tools that we used for T-cell epitopes were EpiVax and IDB consensus method. Again, the important thing to strain is there is no cyno-specific class 2 MHC T-cell epitope prediction tool. You can, in some of the tools, put in sequences of MHCs, but we decide not to bother because we were looking for strong T-cell epitopes, and we had other in silico tools that can also be used. So For B-cell epitopes, we looked at linear epitopes. So this is the IDB, has a Koleskar and Tanganaker. I'm probably butchering that pronunciation. Antigenicity method. Why did I use this? Why did I not use a 3D B-cell epitope prediction tool? So if you take a look at some of the molecules, let's say anti-blank cytokine molecules, and you look at what their epitopes are in the patents that the companies are claiming, if you use this B-cell epitope tool, this Koleskar and Tanganaker, you'll actually get, shockingly, these B-cell epitopes will match very closely to what you actually see, what's claimed by companies in their patents. So I thought that was very interesting. And I didn't see that good of a correlation. Of course, I didn't publish any of this with the other tools. AgriScan and PASTA were used for aggregation-prone regions. AgriScan was used as a very easy, simple tool to use. PASTA wasn't as good. AgriScan results actually were very similar to overlapping with the B-cell epitope prediction tool and the T-cell epitope prediction tool. And then there's a thing called an IDB de-immunization tool. And so you can look at your problematic areas in your sequence, and we selected variants that may reduce what they call antigenicity in that particular tool. We used an internal Lilly tool called Antibody Profiler. So these variants that were chosen from this IDB deimmunization tool were fed into this Antibody Profiler tool. And what this does is it selects only the variants that are most common in human germline to deposition. So you don't want to put in a rare amino acid that probably some human has never seen before at that particular position. And then those selected mutations were cross-referenced against GenBank to select the variants that are only naturally present in non-human primates. This wasn't limited to just cyno. We looked at other species just in case, such as rhesus. So this is what it looks like. There's been some publications before about in silico tools overlapping with each other, and that's probably a hot spot. So I think we referenced that in the paper, but I'm not going to show that here. But you could see, it's not perfect in every single region, but where, Epivac's T helper, B-cell epitopes, and aggregation-prone regions tended to overlap, we focused on that region to try to deimmunize it then. So this is what the deimmunization tool looks like. This is the printout basically from the IDB, and you could see at different positions, it selects substitutions that they predict will actually drop the score, essentially. And so we arbitrarily set this criteria. This wasn't something that they had for, it was basically our own. And so you can see there's positions here, V3T, V3A. Here's the IG profiler tool that Lilly has, and you can see if you, it's very hard to see on the screen here, but V is normally present at position. That's 82% of those sequence in that particular germline family that pops up 82% of the time. And you could see that T is actually is also accepted at this position. So you don't want to use anything that's rare, essentially, because it likely won't probably express. And so we felt it was safe to put V3T in at that position. Then you could also look at antibody structure. We don't want to perturb here in this Collier de Perles map. This is available from imgt.org, but you don't want to perturb the structural features of the antibody. So we don't want to mess around with the canonical regions, Vernier zone, interface residues. We wanted to avoid those because we weren't trying to do like affinity optimization or mess around with stability of the CDRs. So we avoided perturbing those areas, even if they were predicted to have some immunogenicity liability. So the hypothesis was that substitutions that are normally present in naturally occurring germlines and not being disturbed, not rare, should express well. And to our delight, actually, all the naturally present ones from the use of all these tools did actually express very well, very high titers in our expressions. There were a few exceptions, of course, because you're not going to get everything. So here's on the left side is a printout. So final variants were selected for cyno studies from these, and you could see this was just Epivax. Again, this human-specific tool. You can see your starting protein sequence here. Had epimatrix score 45, and you can see the various substitutions. And these are subtle. You're not going to, there's very few point mutations that'll dramatically drop the score, and oftentimes when they do, it eliminates binding. So in the case of that one position on, adalimumab that eliminates binding, it also it has like a 25 jump or drop in the epimatrix score. So it's like one of the largest changes I've ever seen. Of course, these are subtle, but it just takes a little bit. If you have a very clean molecule, you reduce its liability just a little bit, then the tolerogenic T-cell epitopes will hopefully overshadow the ones that are immunogenic. And you can see the other chain here. There's some other reductions as a score as well. Again, the same screening funnel was used. We tried to have as similar to the parental as possible. Of course, better in silico profile, which is what wins out in the end. And these went to a 2-liter CHO transient again. And so now I'm showing here adalimumab. So again, germ lining mutations, as you can see, made a dramatic difference. And so this basically highlights the fact that, maybe sometimes you don't really necessarily need to deimmunize. If you have a really well germline molecule, so really well humanized molecule, that may be sufficient in a lot of cases to actually give you reduce the ADA. Now you can see here, exposure goes out to 840 hours. This was we stopped the study at that point, basically. 2 of the animals did lose exposure after 672 hours, so it wasn't perfect, but it was a good reduction in ADA here to adalimumab. So one of the deimmunized variants here, this middle green figure, you could see all the ADA was eliminated. Now remember, this still binds membrane TNF and antigen presenting cells. This has all the similar features as Humira, so it's binding membrane target antigen presenting cells, yet it's not immunogenic. So that whole hypothesis is probably incorrect. However, what it does highlight is that if you do bind something that's a membrane target antigen presenting cells, you just need to have a much more cleaner molecule to avoid ADA. So the bar is just raised for those types of molecules. And deimmunized variant number 2 didn't work as well. All of them had, of course, the germline wouldn't have to have low titer ADA in order for some of this to be clearing. Same thing with the deimmunized 2, but probably nothing here in the deimmunized variant number 1. The finding was the same for golimumab. The germline variant was very good. You start seeing a little bit of drop off in exposure, 840 hours, showing you didn't eliminate all of the low titer clearing ADA. The immunized variant 1 was also solid. I mean, it was clean. We did detect some ADA by 840 hours, but it wasn't, at least it didn't lead to increased clearance of the molecule. The immunized variant 2 also was pretty good. You start seeing a drop off a little bit in exposure here. So it's not perfect, but basically these have been reduced quite dramatically. So here's a little table of some of the physiochemical properties. Again, these aren't perfect. We tried to maintain similar affinities. There were a little bit of drops in affinities for some of these molecules, a little bump up in affinities for, golimumab, but of course it's never going to be perfect. Fab TMs were very similar across the molecules. Percent aggregates were all really low, and then I have the epimatrix scores here in the columns as well. So we did perform DC internalization. We wanted to see how well do our molecules internalize. Now this is with human DCs. This isn't with cyno DCs, so that's the caveat there. However, these molecules bind both, cyno and human membrane TNF, so this should be fine. So as you can see, we have a control molecule that we use that's normalized to be 100%. And then the other molecules are normalized to it. Interestingly, golimumab is less immunogenic than adalimumab, but internalized faster into DCs or to a higher rate, which is something that surprised us. So again, we present and people talk about different risk factors for immunogenicity. DC internalization comes up. Yes, things that internalize very rapidly are going to be immunogenic, but you have 2 molecules binding the same target. The more immunogenic one internalizes to a lesser degree. So it's not always going to be the case. However, when it does internalize, it has a poor in silico score. So it gets the peptides just get presented better, essentially, to T cells. And you can see the variants all had, roughly similar DC internalization. So we didn't alter DC internalization of these molecules. Here are the PK parameters of adalimumab and golimumab. It should be noted, you have these what appears to be weird half-lives. So for those that are in the know about how PK parameters are calculated, you try to get as many points in a terminal phase as possible. When you have the presence of something like ADA or you have a target-mediated component, you sometimes don't get sufficient points in a terminal phase, or you get this little weird drop-off, and so it affects how half-life is essentially calculated. And so because of that, we try to use something more standardized, such as clearance as an example. And adalimumab behaved very similar as it's predicted to in humans, has been shown to in humans, and previously in monkeys, right about 0.2 clearance. Anything with like a 0.1 to 0.2 range in clearance is a very well-behaved molecule. Golimumab here in this bottom table, you could see clears about twice faster than adalimumab. This is consistent with what we've seen before and what's also published in the submission documents. And you can see what's interesting though is the clearance actually improved for golimumab from chimerization to germ lining and deimmunization. Likely this is due because again, you see an increased clearance here for golimumab, and part of this calculation too is also could be affected by the drop off in exposure and which points you're using, and so you are seeing an improvement in the clearance of the molecule. So to conclude here, so foreign sequences, as I've shown, are not inherently immunogenic, but they do remain a very large risk factor. Binding a target of surface antigen presenting cells is not inherently immunogenic. Sequence quality, rarity, presence of different types of epitopes, and biophysical properties are likely the largest determinants for predicting immunogenicity risk. So, try to have a very clean molecule that looks as human as possible, doesn't have any, bizarre biophysical properties or is prone to aggregation. We've shown here that backbone IgG1 provides some tolerance to the immunogenic V regions. And then, the thing that's just surprising is that just good germ lining techniques may be sufficient in most cases at reducing this risk. And I think for a lot of molecules in the past, deimmunization was not a common thing before, and good germ lining techniques made clean molecules. Of course, there are fully human molecules that are very immunogenic, and this is why you need to look at the in silico hotspots as well and try to remove those and have a good model system for testing it. So. And then, another thing about just using monkeys in general is that outbred strains of cynomolgus macaque are in most cases suitable in view of models predicting immunogenicity risk. And these are the individuals that contributed to the work from Lilly. And thank you for the time to let me talk about it.
Beautiful work. This is it's very cool. I mean, this is such a, I think, changes the paradigm around a lot of the how to address these things, and really start to nail out how do you parse these things out, and how do you put numbers on it, and how do you compare and contrast. Could you go back to the funnel that you had and talk about how you the different numbers and different different sequences you get as you go through that funnel, and what the dropouts are?
Sure. So I think we had, because we wanted to try to fit, so we didn't want to use any type of system that would be like a phage or a yeast display, only because in a particular ADME group, we would have to then send that off to a different part of like Lilly to do studies. So we wanted to keep it internal. And so, I've been humanizing and deimmunizing molecules and fixing them for quite a long time. And so I don't want to say it's like an art or anything like that, but you start learning, you start seeing patterns. And so we thought that we could be smart, I guess, and try to figure out, well, which are the most likely to succeed. So we generated this library. Roughly, it's about, I think it was stopped somewhere around 424 well plates in this for the number of clones. And shockingly, I was thinking, just based on my previous, where I didn't use as many in silico tools as my previous job, I was thinking, okay, 90% of these are all going to drop out just in the expression titer alone. That's a very good indicator for me, just my past experience. If something doesn't express well, it's got inherent liabilities and problems, and you probably don't want to futz with the molecule too much. And so, of course, there could be other reasons for it not expressing well, but generally speaking, for an antibody, if it's not expressing well, so these are full-size antibodies that were expressed. These weren't fabs or anything like that. So, unfortunately, at this first step, maybe 20% of them only dropped out. So we were hoping for a lot more, because it would have made all the biophysical measurements much easier to do, because then you'd have less clones to work with. But because we use that approach of only picking commonly, non-rare residues at these different positions, that they almost all expressed very highly, hundreds of mg per liter, this small scale. I'm getting close to a gram per liter, basically.
Yep.
So similar percent aggregate, again, we put these through, and this is, we lost probably about half the molecules for the percent aggregates. Some of them, they express well. I've seen this before when I did yeast display. You get these molecules that, again, here I'm going to basically downplay what I just said about the expression, but there are cases where there are stable aggregates that form, and then these basically can still express well. So it's rare that it happens. And so we did lose some here in the percent aggregate. And we were splitting hairs. We're talking we're eliminating stuff that had 95% monomer basic, because we had a glut of molecules, like 99 point something percent, very high 90s. And so we said, okay, well, anything that's like 95, we'll just cut off and we just put it away. Yep. So we lost about half of them there. The affinities were also, I'd say probably this dropped most of them. So then we were left to basically like, 3 or 4 molecules. And then these went into DSC, and then it was very easy to then perform in silico. It's more automated now. We have at Lilly, we have an automated system that was built then. But for these particular studies, this was done mostly by hand. And so it was painstaking to take more than like 3 or 4 into the in silico. And then we just selected a couple for 2-liter CHO. And then that's basically how it funneled away.
Yeah, I think this is really interesting. I mean, Sridhar will talk a little bit about how very quickly and how we address the same problem, but it's very cool to see this data.
Yeah, this is pretty exciting, and also offers hope, I guess, that we can deal with this issue. So one quick question I had was, when you predict these binding epitopes using in silico tools, this must be some sort of a cutoff on the binding affinities or IC50s from those predictions, because you do get a lot of them. I mean, almost every molecule, as you said, even human molecules.
Yeah, everything's got them. Yeah,
everything is there. So is that something that you look at certain amount of minimum or like low binders? Or
yes, so there's, if you look through publications and then you look on IDB's website, they have this like little tool that tells you what they think is low, medium, high. And so essentially low is 10% to 5, so 5 to 10% of percent of binders for that particular, however they calculate score in their consensus method is low. There's of course greater than 10% is essentially no risk at all. So we didn't use any of the 5 to 10%. So these oranges basically, or yellows, are somewhere around hovering between like 2 and 5%, and then these reds are much higher, so below 2% of the for the consensus method essentially. And then for Epivax, this is Epivax here, but that's what we did for IDB. For Epivax, we used anything that had what they called their bars essentially. So I think I want to say yellow was. So it was the criteria was very arbitrarily set because of just the way that some of the stuff works, and I think it was I think I called yellow for Epivax if I saw 2 or more of moderate risk peptides. The orange was it has the presence of I think 2 high risk, they're dark blue, and then Epivax is scoring, and then red is 4, which they would consider to be a strong bar. I believe that's how I did it. I'm gonna see if I have. I don't think I put a printout here of Epivax in this particular presentation, but that probably would have been helpful to have.
No, but I think I like the way you use this tool and actually build the molecules and empirically test too. Yeah, so I think that's a key, because you pointed out that a lot of these may not actually be immunogenic.
No, and then there are another thing comes up is you look at framework 3 of a lot of these molecules. So framework 3 in most human germlines actually is considered to be a T cell epitope, even though it's 100% germline. So you kind of have to go over it and with a fine-tooth comb and say, well, this probably is not really real. The tools are all meant to have false positive rates, but they don't want to miss something. And so again, you look at antibody sequence and like half of it lights up with these tools. And so you just have to go back and that's why I think the accuracy of those methods increases when you start using the other tools on top of it. And if something has a strong B cell epitope, a strong aggregation protein region, and it's got T helper epitopes, then maybe you should pay attention to that region.
Yeah. So one other quick question. Each of these had a corresponding germline sequence. So do you compare to see if that epitope already exists in the germline, and do you worry about it, or?
You're exactly right. So I think I have a diagram here. So these are, if it's 100%, so if it was 100% human in a particular germline and it's the same sequence for cyno, yet it's predicted to be a T cell epitope, I didn't engineer it.
Okay.
So I just left it. There's different hypotheses about that, why that is. I have my own hypothesis about why framework shows strong T-cell epitopes and it's 100% germ. I have a feeling that it's some kind of tolerance mechanism, basically, or trying to overpower actual immunogenic T-cell epitopes, and so they're just protected really well. Even if you do maps on these molecules, you will get those regions. We do maps routinely at Lilly, and we see framework 3 is always presented really well.
Yeah.
Unless if it's a rare amino acid that's not normally present, then you need to pay attention. But we feel that likely it's not going to be a problem.
And how well does MAPS correlate with your real world studies? I mean, if you do the same thing here now with MAPS instead of Sino, what would the outcome be?
So we actually did not do MAPS with Sino. That's one thing we would definitely want to do with these molecules. So that's next on our list of things to actually get done. But the MAPS in using human cells correlates pretty well with the in silico too.
Okay.
It just gives you, the in silico tools give you 9 mers, or IDB gives you slightly larger peptides. MAPS will give you like the whole class 2 peptide, essentially, including the core.
Okay, so do you want to pull up your slides?
Yeah, sure. I think this is great. I think
this is we could have spent all day.
I can't.
Yes.
Yeah, I just thought I'll give you a little bit of an overview of what we do here at ATUM. Let me share the slides in a second. I think we implement a lot of these, and I think there's much more to learn. Okay, so you guys can see.
Yep.
All right. So I think, at ATUM, we engineer a lot of antibodies, both for its affinity and neutralization type properties, as well as we humanize, so to speak, as again for the same goal as what you presented it for. And most of the time, it's a murine antibody that comes in and probably already tested for a lot of properties as a chimeric human antibody. But again, I think a lot of our work has been focused on the framework itself. And as you pointed out, germ lining is very critical. So idea is to see which is the fit, which of the germ lines that is homologous and close enough that could be a better fit in terms of presenting the CDRs of the murine antibody. So we use tools, again, computationally to figure out what's a good fit, and then evaluate the grafted sequences or germline sequences and find hotspots. I think, again, we use similar tools like you did, primarily rely on IEDB predictions and identify mutations that could be very critical for reducing the immunogenicity, for example, right? Again, I think one thing that good to see in your slide was the effect of these changes towards is binding affinity or efficacy or activity. You didn't lose a lot, but in some cases we have noticed that there could be a drop, and how can we pick it back up, right? So we may need to restore certain mutations back to the murine ones in the framework regions where we have grafted the germline, and all of those changes, again, you built a nice shuffle library. In our case, we still consider all of those and use our approach to design and build a set to evaluate. So anyway, we use very similar tools. It's exciting to see new tools from your end and validated too. So let me go to the next slide. So our process typically looks like this. We look at germline sequences, we design a set of these, of the order of 48 or 96, and then the next step is to go and make them. These are full-length antibodies, as you were talking about. So it's impossible to do some sort of a high-end, high-throughput screening. So we have to do in a similar way, but what we don't do is to funnel them, but because we have 96, we can measure all the properties. That's interesting, the biophysical properties, the binding affinities and so on, and effectively look at that data, and I'll explain exactly how I kind of give overview of machine learning. Learn from that data. So we take those designed variants and we build models and use that data to see what we learned and what mutations or what grafting or what germline are adaptable, and then make a new set based on that learned data. So yeah, again, we use design part is basically using a lot of knowledge, and that includes a lot of in silico tools, and construct them in a way that we can learn from them. And really empirical validation is done using biology, and the optimization itself is done using machine learning. Yeah, so just a very simple uh, slide here to actually show you what we do in machine learning. Basically, you give the data set, you say you want to optimize some variables, you want to build a model, and you want to provide the empirical data and say, hey, which of these sequences and the variables that are built into the design that has the best effect in producing a molecule that checks all my criterion. So we build a set with various different germline framework, identify various hotspots just like you did. Also look at structure incompatibility mutation. I mean, something can look great, but no binding because it's not presented well. And also look at what mutations that are already present in our, let's take an example of murine sequences that's conserved there, but we would like to change it to human, but may have an effect in its properties. So we put them all together in a bag and then make a set of variants and measure them. So this gives you, again, going back to the slide, a model from which we can then build a new set, or not. I mean, some of them might actually work out well, and we pick the candidate. Roughly how ATUM works with these sequences. So, Klaus, is there anything you want to add?
Now, if you go to the next slide, one out.
This
so,
one, yeah?
Yeah, that one there. So it was really interesting. I mean, what Luca's been talking about is validating more on the immunogenic side, what you see on the Y of the output, right? So here, really looking at this from a real in vivo perspective, which is very cool. I mean, that's certainly something that you know, we got to.
Well, it's going to be hard to do with 96 macaques.
Yeah, well, there's a little problem with that. Yeah.
Well, what your study shows is that a lot of these in silico studies are actually helpful in determining. So that's a very promising thing for using mathematical models to design these sequences.
No, absolutely, and then really start capturing not only the each hotspot in each edit by itself, but really start finding the epistatic effects, right? We use the synergies and antagonistic results coming from the input variables, the X variables, and how they affect.
I mean, there's a lot of examples where if you are screening for binding, it may not actually have the effect in vivo that you would desire, right? So
well, biology is complicated, right? That's but I think that the cool thing really coming out of I was just blown away by your paper when I first read it, is that there's quite a bit of accuracy in those in silico tools, much more so than honestly I anticipated.
Same here,
yeah. Yes, certainly.
So I think that's very promising for the field, and it really makes it clear to us that there is. It's not. I mean, you mentioned that in the beginning. You can go to the thank you slide. It really is more than just voodoo. It's more than magic. We're getting to the point now. We're actually getting some science behind it, which is very promising.
Yeah, and then we're building databases too for trying to predict PK as well, and so I think that's the next. How these different changes or biophysical properties, without going into a monkey. So I think the machine learning is make tremendous leaps in the past few years.
No, it's very nice to see, and it really. I think the biology is very amenable to machine learning because it's very qualitative, right? you either have valin or leucine at the position. You can't have 0.6 valin, or it's very,
Yeah.
it's very qualitative, everything you do. And that means that you can really put proper numbers on it and use that information to drive your machine learning process.
Yeah, very good.
Well, I think I really want to thank you, Lucas, for joining us, and I thought it was one of the best talks I've heard in a long time. So I really appreciate that.
Thank you. I appreciate it. Yeah, it was fun. I enjoyed it.
Yeah, thank you so much.
Thank you. Thank you very much.
Thank you, guys.
Have a nice day.